Okay, so 30 minutes. Great. Good, good, good.
Yes.
Give me one sec to load my slides. Thank you very much for inviting me. That's very kind.
It is pleasure to welcome you for our symposium. We are really excited for your talk,
The
and
pleasure
we
is all
thank
mine.
to your acceptance.
Right. No, there is no better, no bigger honor than having students ask for a talk, and that's wonderful. So tell me a little bit about the audience.
They are mostly undergraduate students. The audience is, I think, there are a lot of medical students, and some are biology students, psychology students.
Great. Excellent. Then I can pitch it right. Wonderful. Good, good, good.
I'm Sara, by the way. I didn't introduce you myself. I'm head of the scientific team, and Sura will moderate your session. She is
Very
here
nice to
now.
meet you. Very nice to meet you. I'm George. Yeah.
It's my pleasure.
Great. Hold on. Loading the slides now, and
Mhm.
off we go. I think I sent you the wrong deck, so hold on for a sec. I'll send you right after my talk. I will send you the correct PDF file. Okay,
Okay.
great.
If you can open it.
Yes, I'll open it right
Okay.
now. Share. Do. That should be it. All
Okay.
right. Can you see my
Yes, we
screen? Great.
can. I let's the talk to you, Sura, now. Um,
Excellent. You see the cursor?
Okay, first of all, yeah, we can see.
And you see the full screen, right? Not the small split screen.
I see two screens right now.
Oh, you see
One
two.
big and
Oh,
the
hold
small
on.
one on the next to it.
That's not good. Let me change this. Um, Give me one quick sec. I'll do it. I will do a very quick display change here. All right. So now you should be able to see one screen, right?
Yes, it's okay now.
Excellent. Good. Great.
Okay,
Wonderful.
so I'm gonna do a little introduction and then if it's okay with you, we can start.
Yeah.
Okay, first of all, hello everyone, and thank you for being part of this session. It is a great pleasure to be here. My name is Sude. I am an undergraduate student at Ankara University, Department of Biology. I will be moderating this part of the symposium. We are very excited to host Professor George Malliaras today. Thank you for taking our invitation and for this symposium. Professor Malliaras is the Principal Professor of the Technology at the University of Cambridge. His research focuses on the organic electronic and bioelectric, especially their application in medicine. He is a member of the Academia Europaea and has received several awards from the European Academy of Sciences and the Material Research Society for his work on targeted drug delivery. Today, he will be presenting on the topic Technology for Bioelectronic Medicine. Moving on to the presentation, I invite Professor Malliaras to begin his presentation.
Great. Thank you very much, Sudh. As I was saying earlier to one of your colleagues, there's no bigger honor for an academic than to be invited by students to give a presentation. I'm privileged and delighted to be with you. So what I wanted to do for the next half hour or so is to talk about the field of bioelectronic medicine and reflect a little bit about future trends. So I'll go through an introduction, then show you some results, and then try to predict what will come in the future. So bioelectronic medicine involves electronic devices that interface with the nervous system in a bidirectional fashion to provide treatment for diseases and injuries that currently cannot be treated with conventional methods. And this is very important as it's a new way to deliver treatments to people who need them. This is one example. This is a patient here who has tremor. Tremor is a movement disorder that's associated with the uncontrollable movement of the extremities and makes life difficult to live. For example, you see they have hard time scribbling something on a piece of paper, holding a drink of water. And these conditions typically are initially treated with some pharmaceuticals, but soon enough, the pharmaceuticals would lose efficacy. So today there is a new way to treat those conditions, which is called deep brain stimulation. And it involves placing electrodes deep in the brain, in the subthalamic nucleus typically, and stimulating that location with electrical current. And immediately you see a significant amelioration of the symptoms. So this type of intervention is being used now more and more in people with Parkinson's disease, in people with dystonias, after medication stops working for them. The implants that are used today are shown here. This is a rather recent summary that indicates the different locations where the implants are placed inside the body, as well as what indications they treat. For example, there are cochlear implants that go in the inner ear to stimulate the cochlea to help people with profound deafness. There are implants that are placed on the brain for people with movement disorders, with some severe forms of epilepsy. Implants, of course, on the heart to help with different cardiac conditions and a variety of other indications. And by and large, these implants look like what is indicated here. There is a box which is called the implantable pulse generator that contains batteries. About 2/3 of its volume is battery. And then a third of the volume is electronics that take the power from the battery and convert it to current pulses that are delivered by an electrode array at the appropriate location. For the case of deep brain stimulation, this is how it looks. So there are two electrode arrays here, one in each hemisphere, the right and the left hemisphere, stimulating the subthalamic nuclei. So the electrode goes into the brain, and then outside the brain, you have this wiring that goes typically under the skin and over by the side of the neck, goes under the skin of the neck to the chest here, where under the skin this little box is placed. Now, when the electrodes are placed in the brain, they stay there for the life of the patient. So typically, the electrodes are implanted once and never removed. This little box usually gets removed every couple of years to change batteries. Typically would last maybe 5, maybe up to 10 years, and then it would have to be removed. That's done by making an incision here on the chest, removing the old box, plugging in a new one, putting it in, and then suturing. Now, this exact example that I show here is indicated as RC. This means rechargeable. So patients who use this particular stimulator, they have a device which looks like a wand that they will put over their chest to recharge the batteries. So that's also a possibility. Now, surprisingly, these implants are still made by hand. We live in the year 2026. We have a lot of technology available to us, but still these medical devices are made using very primitive technology. So you have people working through microscopes with tweezers assembling these electrodes together. This dates back to mechanical watchmaking. That's how watches in the old times were made. And the reason why this persisted in medical devices is because the industry is very reticent towards change. They're afraid that if they change their manufacturing process, they change the materials that go in these devices, the device might fail, and this will make them liable for a lawsuit. However, this is now being disrupted by a lot of small companies that are using the techniques of microfabrication that typically are used to make microchips that go on cell phones, on computers, to make implants. So these devices can be made using traditional processes used in microelectronics, such as film deposition, lithography, etching, to make objects of exceptional precision, reproducibility, and at an ever decreasing cost every year. For the same amount of money, you get a better performance, typically in a computer, in a cell phone. So these tools and technologies are now beginning to impact medical devices. This is one example. This is a clinical case where an implant, an electrode array is placed on the surface of the brain to measure electrical activity. This is a procedure called electrocorticography that is used to map the brain before an operation. So what you see here is a craniotomy. So the scalp and the skull are removed. This exposes the dura mater. This is a membrane that encloses the brain. Then that membrane is cut here. So this is a durectomy. The membrane is pulled back to let the surface of the brain become visible. And then an electrode array is placed here to map the brain. However, as I mentioned earlier, these typical medical devices are made by hand and have very large features. So the electrodes here are 5 millimeters in diameter, and the mapping that they provide of the brain is fairly coarse. Now, with microfabrication, you can miniaturize these devices and have hundreds of electrodes in this area, each electrode being as small as 10 microns. This is significant, 10 microns, because that is the size of the soma of the body of a single neuron. So these devices can map the brain with single neuron resolution. So as you measure the electrical activity here, you see these spikes, and these spikes correspond to individual neurons on the cortex that are being recorded. So the point that I'm trying to make is with this microfabrication technology, you can have very high precision in recording the brain. This is also now in the commercial sphere. This is a company called Neuralink, which makes an implant for paralyzed people. So these implants are in the form of linear electrode arrays, each one having multiple electrodes. Again, very small microfabricated electrodes of the order of the size of a single neuron. These are pushed into the brain, in the motor cortex of people with paralysis, and they can read volition. They can read the neural activity that corresponds to volition for movement. As a result, they can be used by people who cannot move their arms and their legs to collect information from the brain and then move a robotic arm or move things on a computer screen, a cursor to help compile an email or to play chess. Now, These technologies will allow us to do things that cannot be done with the current state of medicine. Let me give you one example. This is a work on spinal cord electrodes. These are again microfabricated electrode arrays that can be placed around the spinal cord. So you can place this array under and over the spinal cord, presenting electrodes all around the periphery. This is shown here. This is the spinal cord, and then the device goes under and over. With these devices, we can map electrical activity as it moves from the brain to the periphery along the spinal cord. So this is implanted along the spinal cord of an animal. By stimulating the motor cortex, we can cause a motor command that comes down the spinal cord and then goes to a muscle, getting it to move. And with this implant, we can see exactly where these commands are trafficked on the spinal cord. So the signal is high when we issue a motor command, ventrolaterally, so on the side and the front part of the spinal cord, shown here. So we can map these so-called motor evoked potentials. Then we can go in and stimulate at the appropriate location and cause movement of that muscle. For example, if we stimulate ventrolaterally with different electrodes, then we can create movements that are quite specific. You can also put these two components together. You can have two arrays along a spinal cord injury, one above and one below spinal cord injury. So then when we stimulate the motor cortex and create a motor command, that can be recorded with one array. And then that information can be passed electrically around the spinal cord injury site to another array that will stimulate the appropriate location. And when you do this, then you can restore motion. So this is the command for movement that is recorded by this array. And then it is passed to another array past the injury that stimulates. And then you can see that the leg moves, the movement is initiated. If you disconnect this interface, this wiring here, then the motor command fails to propagate. It does not elicit any movement because it can't go through the spinal cord because of the injury. And then when you reconnect, movement is restored. So this is a very simple, at this point, demonstration that it points to a future where maybe one day we can help people with spinal cord injury that leads to paralysis by making bypass systems that record and stimulate to enable to bypass the injury site. As I mentioned, again, this is very simple. In this case, we're dealing with an anesthetized animal, and we ourselves are eliciting this motor command by stimulating the motor cortex. In real life, this animal or a person with paralysis would have much more complex activity that might be hard to analyze. So to demonstrate that this works also under more complex conditions, we looked at freely moving animals. We put an electrode array on their spinal cord and then recorded these movement activities as the animal moved about. You see that the signals look very complex, very complicated compared with the previous case in an anesthetized animal, but still they can be analyzed to predict movement of the leg. So what you see here is these markers that a camera is using to measure the exact position of the leg. And what we can do, we can compare the position of the leg as obtained by the camera with a predicted position of the leg as predicted by analysis of this data. And the two coincide. So you see two curves here. One shows the actual position of the leg as measured by the camera, and the other is the prediction after analyzing the complex data that are recorded from the spinal cord. So this is very promising because it points to the fact that we can still make this idea work in more complex situations. Now, finally, I will show you another a last example of what else you can do with this technology, with this thin film implants. Here we are interfacing with a nerve which is called the pudendal nerve that comes out at the bottom of the spinal cord. And that nerve ends up innervating visceral organs. For example, it's the nerve that helps, that conveys information about emptying bowels and emptying bladder, which are functions that are severely affected in people with spinal cord injury. This is a very complex nerve that has many fascicles. And the question is, by recording on this nerve, can we get those signals specifically and then help people with paralysis who have trouble emptying their bowels or their bladders? So we made cuff electrodes, again, that go around the pudendal nerve and present different electrodes. Then what we did is, in an experimental animal model, we used the catheter to distend the bladder. That means to add liquid to the bladder and make it bigger. And that causes signals to go towards the brain. And these signals indicate to the brain that the bladder is full. These signals are severely disrupted in people with spinal cord injury induced paralysis. So what we see here is recordings of electrical activity from the implant that goes around the pudendal nerve. And you see that this information about bladder distension is trafficked at particular locations in the nerve. And these are different locations than if you do, for example, bowel distension. And those are at different places along the perimeter of the implant. And likewise, if you just gently brush the perineum of the animal, you get yet different channels to activate. You can classify those signals and be able to tell in a freely moving animal what the animal is doing in very high degree of accuracy. So you can tell whether the animal is moving, is grooming, or it's voiding its bladder. More importantly, you can elicit these functions on demand. So here you see a video where we stimulate specifically in this red region and here to cause emptying of the bladder. So every time we stimulate in this specific region, you see that the animal opens its bladder. And turns out this happens when you stimulate particular channels around particular electrodes around the pudendal nerve. Here, for example, the electrode number 4 empties causing of the bladder. And it's a different electrode that causes emptying of the bowels of the animal. So we can independently control bladder and bowel functions in a freely moving animal. This is very promising news for people who suffer from spinal cord injury, as these functions are severely disrupted in several cases. And here we show a technology that can help restore those functions on demand. So where does all this go? The experiments that I showed you today were with animal models, and we are taking the translational pathway to bring them to the clinic. So you start with small animals, then you go to large animals for a safety study, and then you go to the clinic, and they are taking their pathway towards the clinic. But if I look at research in this field, where does it go? Currently, with bioelectronic medicine, what we do is we stimulate existing neural networks inside the body. These neural networks are often compromised by disease or injury and often further degrade by neurodegeneration. So it is quite limited what we can do with just simple electrical stimulation. In the future, I foresee a technology where we combine implantable electrodes with cells and put them in the body at the same time. This would help rebuild the compromised neural networks inside the body, and then we can control networks that have been repaired. There is a lot of evidence in literature, for example, that electric fields can help cells migrate in a preferential direction, can help stem cells differentiate to different fates, and can even help cells sprout out new projections. For example, they can sprout out different axons, which can help connectivity inside the body. So to demonstrate this point, we worked on a peripheral nerve injury model where we implanted on a severed nerve a device that had muscle cells and electrodes. So these cells were differentiated into muscle ex vivo. We started with induced pluripotent stem cells cultured on a device. And then after 10 days, they had differentiated into muscle that we then placed in contact with a severed nerve. And what we found is that if you do this, if you put a device with cells on a nerve, the nerve will innervate the cells. So then when we stimulate the nerve, we can measure the conduction of signal by our electrodes. So these are electrical activities from different electrodes that show that this severed nerve is now being recorded by our electrode array. If we do not put any muscle on the implant, if we just take the implant and put it on top of a nerve, then we don't measure any activity because this part of the nerve has degenerated because of the injury. So in a sense, what I'm showing is that the co-implantation of electrodes with cells enables a repair of the nerve. And then you can also monitor this repair process. This is recordings from one electrode in week 1, 2, 3, and 4 after implantation. And you see that the signals take a little bit of time, take 4 weeks until the nerve is repaired and you start recording good signals. And once this happens, then you can record volition of the animal to move. Every time the animal is moving, you can pick up electrical activities that correspond to that motion. So this is where I think the future of this field lies. So to conclude, I hope I gave you a flavor of what bioelectronic medicine is. It's a new and emerging field where we use implantable electronic devices to interface with the nervous system, and that helps to treat disease and injury in new ways. We talked about technologies that are inspired by microfabrication that is used to make electronic devices. These devices now, this technology now is used to make implants that give you very high precision. And I showed you two examples of that. One is new uh, spinal cord injury bypass that allows us to bypass along an injury. And the other is peripheral nerve system interfaces that allow independent control of autonomic function. In the future, what I call bioelectronic medicine 2.0, the co-implantation of electrodes with cells will enable not only to stimulate existing cells, but to repair networks that were damaged by disease or injury, and then interface with that. So with this, let me thank the group. The group is co-led by a neurosurgeon. I'm an engineer myself, and has group members that range from engineering all the way to medicine and surgery. Collaboration with people in the university and outside, funding agencies here. Thank you very much for having me. It's a pleasure to be with you.
I'd like to thank you for this insightful talk. It was a pleasure having you with us. Now,
Thank
if you
you.
have a few more minutes, we'd like to ask you some questions from participants.
Yes, very happy to do that.
Okay, our first question is going to be, how much do you think this technology would be developed? Do you think that it would be possible for this technique to solve most of the severe brain disorders people are struggling with?
Well, it's not very clear at this moment. So one of the biggest neurological conditions is dementia. It affects a lot of people. And it's not clear how bioelectronic medicine will help people with dementia. The reason is that dementia is highly delocalized over the whole brain. Where bioelectronic medicine has been successful is in conditions that are localized at a particular location. For example, in Parkinson's disease, we know that we have to put electrodes in the subthalamic nucleus. In epilepsy, we know that we have to put them in the epileptogenic zone. So although it currently has a pathway for treating a broad array of conditions and injuries, it's not clear for things such as dementia where exactly you would intervene. So it's still a bit open. There is a lot of research to be done to understand these issues.
Thank you. We have another question. Do you think this technique could be developed for other regions other than the brain and the nervous system?
Yeah, so there are typically bioelectronic medicine started out of the heart. So people were treating disease of the heart, such as arrhythmias. Then that was in the late 50s or so, where the first implants for the heart were the first pacemakers. Then it went to the inner ear for helping people with deafness. These are the cochlear implants. The first ones were developed in the late 60s. Then spinal cord and brain for neurological and neuropsychiatric disorders. But what we see in recent years is an expansion towards autoimmune conditions. So these are implants that are placed on peripheral nerves, typically the vagus nerve or some other peripheral nerves, and can stimulate organs such as the spleen to produce different let's say, down regulate the production of pro-inflammatory factors and help people with rheumatoid arthritis, or on other locations in the body to help people with type 1 diabetes, Crohn's disease. There are a couple of technologies being developed for to go beyond neurological disorders.
Thank you for answering our questions. It was truly very inspiring and very informal. Before we finish, we would like to present you with a certificate. We will also be sending it to your email account.
Great. Thank you very much. And I will send you the slides in a moment.
Thank you so much.
A pleasure. Take care. Have a good day. Bye.
Bye.